Welcome back to Meaning from Data, Statistics Made Clear. In this lecture, we're going to be talking about science and applications of science, statistics to science. Certainly statistics and the statistical analysis of data are obviously central players in all aspects of science. And during the last 400 years, we humans have fundamentally altered our conception of the universe and our position in it, in many cases as a result of a scientific advance that ultimately is based on the analysis of data. So let me first talk about the concept of the solar system. In the year 1600, Johannes Kepler was in the observatory of Tycho Brahe and was looking at the data associated with the positions of the planets as they moved in the night sky. Tycho Brahe had accumulated the best observational data. This is before the invention of the telescope. He had the best data that had ever been collected. And one of the anomalous challenges was to explain the funny behavior of the motion of the planet Mars, which had this retrograde motion and other anomalies that made it difficult to explain. Kepler took the data from these observations and basically fitted a curve to the data to make his laws of planetary motion, namely that the planets revolve around the sun in an elliptical form. So Kepler's contribution to our understanding of the solar system and our place in it was a statistical one. It was data fitting, finding a model that approximated the data that was actually observed. Later, our understanding of the reason for having elliptical orbits was put on a different foundation when Newton proposed his universal law of gravitation, from which it follows as a mathematical consequence that planets that were undergoing this kind of inverse square law of gravitational attraction would in fact automatically follow elliptical orbits. So that changed the presentation and our concept of the reality from a statistical curve-fitting concept of Kepler to a one based on a more fundamental physical law. This is frequently the way science proceeds, that at one point we have an observation that's a rather empirical kind of observation that later is put on a firmer foundation when a theory that implies that foundation is found. In our concept of the entire universe, we have the example of Hubble's redshift, which was a statistical observation that there were differences in the spectra of stars that were further away that implied that further away stars were moving faster. There was the concept of the discovery of the 3-degree background radiation that was discovered when it was found that in trying to create a very precise radio telescope, the people were unable to tune it properly to the level that they thought. No matter what direction they turned it in the night sky, they discovered this background radiation. That was a statistical or a measurement issue, which was then later explained as the leftover from the Big Bang. And so it was confirmation of the Big Bang theory of the universe. So measurements are used in both finding out interesting things about the universe and also to confirm a model or reject a model. For example, Einstein's theory of general relativity was confirmed, or we should say Newton's theory of the universe was rejected when observations were made about the light bending around the sun. In order to say that this would not have happened, the measurements that were obtained would not have been obtained had Newton's theory been correct. In physics, the concept of the most fundamental understanding of the basic particles that we live in are probabilistic in nature, live with or made of, are probabilistic in nature. Quantum field theory implies that we are not to think of a subatomic particle as being in a particular place at a particular time, but instead it's a probability distribution, and that is the actual reality. And this very strange idea puts the concepts of probability and chance as fundamental players in our understanding of the physical world. Measurements and the interpretation of measurements are very basic to the scientific process. And I want to give an example where measurements had an interesting wrinkle to them. Back in the 1970s, people began measuring the thickness of the ozone layer in the stratosphere or the upper atmosphere, and I wanted to tell this story as an illustration of some part of a statistical practice. When measurements were made about the thickness of the ozone layer, there were many measurements that were very similar to each other, as you would expect if you were measuring something where the concept was that the thickness was uniform. Many of the measurements were the same, but there were a few measurements that were much, much smaller, that were close to zero. And there was a computer program that was taking these measurements, and it was set to identify measurements that were too far out of the bounds of the preponderance of the evidence and label them as potential outliers. Outliers are, remember, data that don't seem to fit the pattern of the remaining part of the collected data. And so by noticing that there were these measurements which were close to zero, the computer program threw them out as potential outliers. Now, it actually kept the data and claimed, and the data were recalled later, so that was important. But the issue is that we can, in collecting data, very often there are errors that just occur accidentally. For example, our measuring instrument may have been on the blink that day and got a value that was way off. Before we had an understanding that there was an actual hole near the South Pole and near the poles in the ozone layer, it was just viewed as the hypothetical image of our concept of the ozone was that you could imagine that there was a uniform distribution of the ozone layer across the whole Earth. So one question that we have about data is, what is the application of the generality of the application of the data that we collect? In other words, if we collect data from various spots on the Earth and it was pretty uniform, suppose no data had been collected around the North Pole at all, around the poles at all, the South Pole at all, then it might have well been that we would claim that the data implied that there was a uniformly thick layer of ozone across the whole Earth. But the fact that there was this anomalous outlying data, which we then threw out, turned out to be actually a real phenomenon. And we shouldn't have thrown it out. And it would actually pointed to a phenomenon that is actually real. I'm trying to make the point that sometimes the question of the applicability of the data we get is not clear, how generally applicable it is. Suppose that we assume that all fish are the same with respect to some property, then we might do an experiment that just used, say, a salmon, and then draw conclusions about all fish. And that's certainly fine if all fish really are the same with respect to that property, but it's not always clear that that is the case. There could be some lurking variables that make a different species of fish behave differently. So, in any case, science proceeds by developing models of what we think the world is like, and then looking for data that test the models by comparing the experimental results with the predictions of the model. And in many cases, the scientific theory is tested by comparing experimental results to the prediction of such a theory. And one famous example of that is in the case of Mendel's famous experiments with peas. In the middle of the 19th century, Gregor Mendel was a monk in a monastery, and he conducted many experiments that are now viewed as seminal experiments with respect to our understanding of heredity and how characteristics of the parents are passed on to the children. In particular, he had a model in mind where he envisioned that each of the parents had two genes, of which they contributed one to the daughter plant, and then those two genes were made up the genetic basis for the actual appearance of the plant. So what I'm going to be doing now is to explain some of Mendel's experiments and try to describe how it is that those experiments proceeded. So we'll begin with this chart. So here's an example of a typical kind of experiment that Mendel performed. The peas that he was working with had actually many different characteristics that he worked with, but I will just talk about one characteristic, namely color. He also talked about things like whether it was a wrinkled pod or a smooth pod and other kinds of characteristics, but I will just talk about the two different colors, yellow and green. And what Mendel did, the basic concept of his experiment was this. He imagined that he had some plants that had two yellow genes, and he took these yellow genes and then he crossed them with plants that he assumed to have two green genes. It turns out that yellow is the dominant gene, so that if you have a plant that has a yellow and a green gene, or has two yellow genes, then the plant will appear yellow. So when he crossed plants that had both yellow genes, and this is called, by the way, homozygous, meaning that both of the genes are the same, when he crossed a homozygous yellow gene plant with a homozygous green gene plant, all of the offspring had one of the genes from this parent and one of the genes from this parent in its genetic makeup. So all of the offspring of these plants had one yellow and one green gene. And all of them appeared yellow because yellow is the dominant gene. Now, the interesting part of the experiment occurs at the next generation. Suppose that we take plants that were the result of that previous breeding, and we had plants, each of which was heterozygous, that had a yellow gene and a green gene, and we combined them together to form the potential offspring that they could have. The concept that Mendel, and the theory that Mendel proposed, and that these experiments were intended to support, would predict that for each parent, it would randomly contribute one or the other genes to the daughter plant. So there are four possible things that could happen in this kind of an experiment. First of all, you could have both parents contributing the yellow gene, or the first parent could contribute green and the second yellow, or the first yellow and the second green, or both of them could contribute green genes. Only in this corner here, the plants that have two green genes, would the plant actually appear green. This is the pod of the pea plant. It would appear green only if both of its genetic contributions from the parents were both green genes. Now, so the way that these experiments proceeded was that Mendel crossed a bunch of heterozygous plants and looked at the proportions of the offspring plants that were yellow and the percent that were green, with the expectation that in fact a quarter of them were green and 3/4 of them were yellow. That was the expectation, that he was trying to prove this theory by getting evidence by doing these crossings. And among these three, he expected one of the 1/3 of these to be homozygous with yellow genes, and the other 2/3 to be heterozygous, one green and one yellow gene. Now, let's look at some data. Suppose that we had done an experiment in which there were 200 plants expected in each of these quadrants. Then we would expect that in doing this experiment many times, which he did, he would have expectations of this kind of an outcome if this were the size of the experiment. However, you might ask the question, how does he know whether a plant is heterozygous or a plant is homozygous? Well, what Mendel did is, and he specified his method for determining whether the plant was homozygous or heterozygous, is that he took these plants that were in this quadrant, these three, that is, they appeared yellow. And he bred them with themselves 10 times. By breeding them with themselves, of course, if it's homozygous, on each of those 10 times, he would always get a yellow plant. However, if he has a heterozygous plant, he reasoned that the chances were very good that if in 10 breedings, one of the self-breedings would contribute both green genes and the plant would come out green. So that would be an indication that the plant that he started with was a heterozygous plant, that it had the green gene as well as the yellow gene. So his method was to take his plants and do 10 self-cross breedings, and on that basis classify the plant as being heterozygous or homozygous. Now this is this was the data that he collected. He collected a great deal of data which all supported his theory. In many instances, he received in data of this size, he would find that there were 201 plants that he had classified as being homozygous with yellow, and that the ratios were appropriate, very close. In 1936, Ronald Fisher, you may remember Ronald Fisher. Ronald Fisher was the statistician of the lady tasting tea fame. In 1936, Ronald Fisher wrote a paper in which he investigated Mendel's data. And he made some observations about the quality of Mendel's data. In particular, he noted that Mendel's data was too good to be true. Remember that when we are dealing with a random process, we don't expect the answers to always be exactly according to expectation. We expect a distribution of the answers, that most of the time the answers will be within a certain distance of the expectation, but a certain fraction of the time we would expect to have outliers. We would expect to have unusual occurrences, rare occurrences. So one of the things that Ronald Fisher pointed out were that the number of experiments in which Mendel's data was very close to expectation was too great to be believed. So let me give you an example of the reasoning that Fisher used. Suppose that you take a coin and you flip it 1,000 times. You know that on average, that the mean of the distribution of the flips is going to be 500. But you also know that if you actually flip a coin 1,000 times, that often the number of heads will be less than 500 and more than 500. And several times during these lectures, we have seen the distribution of the number of heads, for example, that would be likely to appear if you did the trial of flipping a coin 1,000 times. This is the distribution. In other words, this tells you that occasionally you would get 540 heads, but the proportion of times you'd get it would be very rare. And most of the time, you would have some number of heads in your 1,000 flips that lies somewhere in the big part of this curve. However, this very thin region in the middle here is a region which contains only a small fraction of the data, about 7.5%. And most of the time that you flip a coin, it would not lie between 499 and 501. Most of the time you would get values that differed from 500 by some bigger amount. What Fisher did was to notice that Mendel's data tended to give more outcomes that were within one standard deviation of the mean than would be expected. You would expect outcomes of an experiment that involved random chance to lie within one standard deviation of the mean in a normal distribution about 68% of the time. But that means that about 32% of the time you would expect the results of that random experiment to have values outside of one standard deviation from the mean. And yet, it turned out that the data that were reported by Mendel had too high a frequency of good data, of the data being too close to expectation. So Fisher was arguing that the data was not properly constructed. But Fisher went on to make another interesting claim about the results from Mendel's data, and that is the following. If you recall, the strategy by which Mendel chose to classify a plant as heterozygous was to do the experiment of self-breeding 10 times. And if the plant came out yellow all 10 times, it was classified as homozygous. There is a chance, a small chance, that if one took a heterozygous plant and by randomness alone bred it with itself 10 times, that every one of those 10 times it would have contributed a yellow gene and would be yellow every one of those 10 times. And in fact, it's not a difficult computation to see exactly what proportion of the times that would happen. Namely, the chance of when you crossbreed a heterozygous plant, that is a green and a yellow plant, and you randomly pick a green and a yellow, you randomly pick the two, the chance is 3 out of 4, 1, 2, 3 out of 1, 2, 3, 4, that one of the two genes contributed, at least one, will be yellow. So there's a 3/4 chance when you when you cross a heterozygous plant with itself, there's a 3/4 chance that it will be yellow. If you do it 10 times, there's a 3/4 to the 10th power chance that it will be yellow every single time. 3/4 to the 10th power is 5.6%. So the probability of misclassifying a heterozygous plant is 5.6%. In other words, if you use the method that Mendel did use of saying, I take a plant, is it homozygous, that is having 2 yellow chromosomes or 2 genes, or does it have a green and a yellow? He bred it with itself 10 times. If it was always yellow, he classified it as homozygous. But 5% of the time that you started with a heterozygous plant, it should have been misclassified as homozygous. What that means is that Mendel should have, in doing his experiments in the way he specified that he did them, he should have misclassified 22 plants that were in fact heterozygous. He should have classified them as homozygous. So what Fisher pointed out was that the actual expectation for that square of the classification of plants should have wrongly classified 22 extra plants as homozygous that actually were heterozygous. So that the actual expectation from the experiment should have been 222.5, not the 200, which was the actual expected outcome that are really homozygous. The effect of this is that when Mendel reported an experiment, and this is a specific example of one experiment of many, in which the reported number, 201, is very close to the quasi-expectation of 200. But you see that it's rather distant from what should have been expected, including the falsely classified heterozygous plants that should have been falsely classified as homozygous using that method. Well, one can do a statistical analysis where the null hypothesis is that the fraction of the plants should have been 222.5 out of 600, and the alternative hypothesis is that the fraction is less than that. You find that the p-value for that hypothesis test is 0.037. So if you use the traditional cutoff margin of 5%, you would actually reject the null hypothesis that the 222.5 was the actual expected value, but it should have been the expected value. Let me quote from, so this is the kind of reasoning that Fisher used in his article to show that the data that Mendel got were too good and were, in fact, in this case, Mendel wasn't subtle enough to realize he should have been expecting 222.5 instead of expecting 200. in that box. And so his data came out more like the 200 than what he really should have found. So I wanted to just quote a few lines from this paper by Ronald Fisher. It's titled, "Has Mendel's Work Been Rediscovered?" And it was published in 1936 in the Annals of Science. And I did want to read that the description of Fisher. He is the Galton Professor of Eugenics at the University College London. By the way, that's another interesting wrinkle to Fisher, which we will not talk about now. But what he said in this paper is the following. The discrepancy is strongly significant, and so low a value could scarcely occur by chance once in 2,000 trials. There can be no doubt that the data from the later years of the experiment have been biased strongly in the direction of agreement with expectation. So he's saying that the data were cooked. One natural cause of bias of this kind is the tendency to give the theory the benefit of doubt when objects such as seeds, which may be deformed or discolored by a variety of causes, are being classified. Such an explanation, however, gives no assistance in the case of the tests of gametic ratios, which is what we were just talking about, and of other tests based on the classification of whole plants. The bias seems to pervade the whole of the data. This is a quotation from his work. I'll give one more quotation. Although no explanation can be expected to be satisfactory, it remains a possibility, among others, that Mendel was deceived by some assistant who knew too well what was expected. This possibility is supported by independent evidence that the data of most, if not all, of the experiments have been falsified so as to agree closely with Mendel's expectations. So this is a little wrinkle in the history of science associated with data being too good to be true. I want to close this lecture with one other completely different kind of experiment that's a little on a lighter tone, and that is that here is a basic scientific question that you maybe have wondered about. How can people count tigers that are in the jungle when they can't find all the tigers? How would you go about counting tigers? There's a very clever way of counting tigers in a jungle, and this is the way you do it. You capture 50 tigers, and then you put ear tags on those 50 tigers, and then let them back into the jungle. And then you let them mix around for a while, and then you come back and you capture some other group of tigers, perhaps 100 tigers. And you see what proportion of the newly captured tigers have ear tags. Then the computation is very simple to get an estimate for the number of tigers in the jungle. So here it is. So we've tagged 50 tigers, so you imagine 50 tigers going around the jungle randomly. You randomly capture 100 of them. Suppose, for example, that 8 of the 100 that you recaptured were tagged. Then you would have this little formula here that 50, the number of tagged tigers, is to the whole population as the number recaptured who were tagged are to the number that you actually captured. So in other words, you captured, among the 100 that you captured the second time, 8 of them had ear tags. So if 8 out of 100 had ear tags, you'd expect the whole population to reflect the same proportion. So just doing the math here, we see that the population would be estimated to be 50 times 100 over 8, or 625 tigers in the jungle. So I always found this a very clever method. It's a question of capturing, and then you let them out, and then you capture some more, and you see what proportion of the newly captured ones have the tags, and using the proportions, you just figure the whole proportion is reflective. You can do the same kind of an experiment with finding the volume of water in a lake. You could take 1 pound of salt in this clear water lake and dump it into the lake. And then wait till it sort of stirs around, maybe several weeks, till it's completely evenly dispersed throughout the lake. Then take 1 cubic foot of water from the lake and boil off all the water until you just measure the salt, and you very, very fine measurement of the salt. Suppose you have 1/1,000,000th of a pound of salt in that 1 cubic foot of water, then you would infer that there were 1 million cubic feet of water in the whole lake. So you can imagine doing this sort of capture-recapture method in many instances. For example, if you had a natural gas deposit under the ground and you want to know how big it was, well, you could put in a certain volume of some number of molecules of something that mixed around, and then if it mixed around, then you could take out a sample from the natural gas and see what proportion of the molecules were in that volume of the second sample and deduce what the volume of the whole find was. So we've seen then in this lecture several examples of statistical applications in science, of which, of course, there are many. This is just obviously the tip of the iceberg in this area. I look forward next time to talking to you about the use of statistics in discovering the authorship of papers.